Hello students, this recording is for Biology 1406, first chapter in the unit on DNA structure and function and gene expression. Chapter 14, open stacks. I think this is fairly self-evident. An interesting approach to the early identification of DNA. He didn't know he was looking for DNA. He was just attempting to answer questions about the composition of this material found in hospital bandages. This experiment is quite fascinating as it was a search for the controlling molecules that theory proposed characterized living cells. So Griffith, the researcher, was working with Streptococcus pneumoniae, as it says right here. And as hopefully you know, most types of bacteria have different strains, which is kind of the same thing as a subspecies. And you know, the more substantial differences in strains are what we refer to as different species in bacteria. They don't reproduce sexually, so they don't have the same genetic dynamics that something like us does. So there are two strains that he identified. The R strain, as it says right here, they're the colonies when they were grown on an agar medium had this rough sort of variegated margin to the colonies, and if introduced to a living organism, they were didn't cause disease. They caused no pathologies. Whereas the S strain, smooth colonies, as it says right here, created pathologies, in an organism when it contaminated that organism, and indeed was deadly. So pathology is there, something that can be diagnosed in a medical setting. So in this case, he injected mice with one of the strains or the other strains and identified them as either being pathogenic or non-pathogenic or as pathogenic. So the crux of this experiment is that he sterilized his S strain by killing them, through enough heat to destroy the bacteria, kind of like the fixing process we used in gram staining. And then he mixed that with live R strain bacterial cells. That mixture, when injected into a mouse, caused the mouse to die. So the R strain, even though being non-pathogenic, was somehow converted into a pathogenic form, and the hypothesis is that it's some agent that was present in the S strain of the Streptococcus pneumoniae. And then, to make it even more interesting, after the fact, live S strain bacteria were recovered. So that means that either the heat-killed S strain bacteria were reanimated, which is, even at the time these experiments were done, would be considered rather remarkable, or equally remarkable, the R strain had been converted somehow into the S strain. And reasonably, he concluded that something had been passed from the S to the R strain. And what we would say at this point, and what he was theorizing, is that it was the molecular basis for the identity and behavior of a particular type of cell. So work in the 1940s showed that the material in question was almost surely DNA that was inducing this transformation event, converting the R strain into the S strain right here. All right, some other very interesting experiments. This one's particularly interesting. Hershey and Chase did a very, very clever experiment working with viruses that infect bacteria. So those are called bacteriophages. And these bacteria, there's lots of different varieties of them that prey just on bacterial cells. So bacteria have their viral pathogens just like eukaryotic cells, including us, have their viral pathogens. And so what happened is they set up situations where they had a radioactive isotope of sulfur and radioactive isotope of phosphorus available in the mineral nutrients that were used to raise bacterial cells. And so radioactive sulfur would be incorporated into any molecules that had sulfur in an experiment, or radioactive phosphorus would be incorporated into molecules that had phosphorus. And when we say radioactive right here, we mean, mildly radioactive, and that they give off photons that can be detected by X-rays. So you can identify them with X-ray plates. You just put the X-ray up against something and wait for particles coming out of that radioactive material to hit the X-ray plate and create a blob of light. So it's kind of a neat technique. And then bacteria, viruses that had been developed by infecting bacteria raised in either of these two events right here, those viruses were transferred over to fresh, naive bacterial cultures that did not have radioactive sulfur or radioactive phosphorus present. Those viruses were allowed to attach to their hosts, and then quickly, as it says right here, the phage coats were knocked off with of the host with a blender. And the idea is that bacteriophages, like a lot of viruses, the protein capsule on the outside of the virus attaches to the target cell, and one way or another opens up the membrane, cell wall and membrane, or just in the case of bacteria, and allows the material inside the bacterial to introduce itself into the cell. So the deal here is that proteins include sulfur but not phosphorus, and DNA includes phosphorus but no sulfur. And so what they found was that the only radioactive material moving from the virus is either the radioactive phosphorus-dosed bacteria in one experiment, viruses in one experiment, or either the sulfur-tagged radioactive viruses in a parallel other experiment, different treatments. Only the phosphorus was introduced into the bacterial cells, indicating that DNA is the genetic material and not proteins. And the reason this is an interesting experiment, A, it's very, very clever, and it did identify what the material injected by a virus into a host cell is, at least for the case of bacteriophages. But it also answers what was a fairly active question at the time. Is DNA or protein the information storing material of a living cell? Both were candidates. A strong argument for protein being the information carrying cell is that it's a more diverse molecule. There's 20 monomers in most living things. There's actually 21 out there if you count bacteria and eukaryotic cells. There's one that's used in bacteria that's not used in eukaryotes. And so you can make a lot of different words with 20 different letters, as you can well imagine. Whereas DNA only comes with 5 different bases, 4 in DNA, and one novel one that's used instead of one of the monomers in DNA for RNA, U instead of T, and uracil instead of thiamine. And so there's a lot of reasons to think that protein would be a more effective way of storing information because of the diversity of letters in the alphabet, if you would, a 20-letter alphabet instead of a 4-letter alphabet, or at most 5-letter alphabet like you see in DNA. But as it turns out, DNA is the genetic material. So this is an image of that experiment right here. So the glowing color represents, radioactive material. So in case you're not remembering it, if you look at the structure of DNA, there's a phosphate group, a sugar, and a nitrogenous base for each letter of the DNA. So there's lots of phosphorus in DNA, but no sulfur at all. None of those molecules include sulfur. Whereas there's several proteins and there are a couple of amino acids, and a very large number of different proteins include, amino acids with sulfur in them. It's routinely used for tough structural proteins, like the capsid of a virus right here. So that was a good thing to identify, a clever thing to identify. And again, when infecting the bacteria with the viruses and mechanically separating the capsid, the protein structure, from the infected bacterial cell itself, centrifuging and transferring those bacteria to a new environment, what we found was the bacteria that included radioactive phosphorus, transferred that material to new viral particles that were produced by these bacteria and were subsequently identified. This was a very interesting experiment by Erwin Chargaff. He did very studious analyses of the composition of DNA in different groups of cells in different organisms and different tissues. And he consistently found that we had adenine, thymine, cytosine, and guanine, A, T, C, and G, in all organisms, and no other nucleotide monomers except for the uracil, which is a sort of an isomer of thymine right here. Slightly different, just a little bit of a different structure, one functional group different. And his primary work that we remember him for is that he quantified the amount of the 4 different bases and found that adenine and thymine were always about the same ratios. If there was 10% adenine, there was going to be 10% thymine in a sample of DNA. And the same thing for cytosine and guanine. They always had roughly the same ratios. And then if you added those ratios of each of these 4 together, that would add up to 100%. So if we had 10% adenine and 10% thymine, that's 20% of the total. It turned out that the other 80% would be equally divided between cytosine, another 40, and guanine, another 40, as an example. And that if you did that for cells of a given organism, you consistently got the same ratios. So 10 to 10 and 4 to 4. In the hypothetical example I just gave you, would be consistent for a particular type of cell, a particular organism. But if you look to another organism or another species of organism, you might find a slightly different ratio, but again, it was consistent from cell to cell to cell in that particular species. And so those are what we know as Chargaff's rules. So the percentage of adenine inevitably matches within experimental limits, error limits, the percentage of thymine, and the percentage of cytosine equals that of guanine. And if you add all 4 together, they'll always be for 100% of your total sample. You should remember the structure of the nucleotides. You need to know all of this fairly exactly. Nucleotides come in 3 different versions of the monomers, by the way, and this is very important in a lot of biological functions. So there's always a nitrogenous base. These are the 5 choices. 4 of these that you find in DNA, and then 4 of these that you find in RNA. U, G, A, and C in RNA. C, T, A, and G in DNA. So this is T and U are the variables right here. DNA for thymine, uracil for RNA. Anyway, one of those monomers is attached always to a pentose sugar. All right. In DNA, that's going to be deoxyribose. In RNA, it's going to be ribose. So there's a hydroxyl group right here, or it's just a hydrogen right here in the deoxyribose. The oxygen is gone, hence deoxy. And then that ribose sugar, whichever version it is for the type of molecule, is attached to a phosphate group. Well, when you find them singly inside cells, they also come as diphosphates and triphosphates right here. And those 2nd and 3rd phosphates represent the phosphorus that powers the attaching of one of these monomers, the nucleotide plus sugar plus a phosphate, to another and to build a chain of DNA. Here's what pays for the polymerization process. It's an endothermic reaction. It is a anabolic bit of metabolism, requires an energy source, and these 2 extra phosphates right here are the energy source for that process. Some other very important experimental work went into understanding the structure of DNA, and the 3 people we want to remember particularly are James Watson, Francis Crick, and Rosalind Franklin. Rosalind Franklin did early work looking at what's called X-ray crystallography of DNA, and that's where you get, under very controlled circumstances, DNA molecules or any other molecule you're interested. You can use proteins as well, and cause them to form crystals as you evaporate the water, the solvent that they're in, and replace that with something else. You get a crystalline sort of condensation of that material. Kind of think of slowly evaporating the water from, some sugary water, and sugar crystals will start precipitating and forming expanding crystal structures at the bottom of the glass. Same idea with proteins and DNA. You can do a similar sort of process. A little more complicated, but you get the idea there. And then if you blast X-rays through those crystals, you get information, if you know how to read the resulting image, that tells you what the arrangement of atoms is in that molecular structure. So blasting X-rays through a DNA crystal gives you this image right here, which kind of looks like a figure 8 right here, and then some rings, and you can see there's a concentric sort of pattern going on here as we go around right here. So what's happening is literally, let me pause this. All right, so if we look at this image right here for a second, here is a side view of the DNA helix. It's a little blurry, I'm sorry, but it's the only picture I could find easily that had both of these views at the same time. So this is the double helix spiraling around, spiraling around. Let me, give you another image that makes that a little bit clearer. So here's the double helix spiraling around and around and around and around. So that's what we're looking at in this image right here from a side view. And then here's what we would see if we were at the end of this molecule looking all the way down the molecule right here. Here would be an end view, okay? And recognize that this is, spiraling around in a particular direction, looking down from above. It spirals away from you in a clockwise orientation. So as we look here, this is going away, further and further and further. And then the next loop down underneath the way. Anyway, that's the idea. And so Rosalind Franklin's X-rays gave this picture right here, which you can decipher that appearance here. So what's happening is here's the two chains right here, very close to the source of X-rays. Very far away from the X-rays and close to the paper, close to the X-ray paper right here. And the X-rays would be coming straight down this picture, like from your eye, straight at this picture right here. And then when they hit these molecules in the center, bouncing off, in any direction around right here, and then hitting the photo paper. And then as we get further and further away from the paper, so going closer and closer to the X-ray source, that angle gets bigger and bigger, and we get these repeating rings right here. So here's the next ring of DNA, the next ring of DNA, the next ring of, and these rings further and further out. And these structures right here represent the bases, or the nucleic acid monomers themselves. And each of these concentric rings represents a revolution further and further away from the source of X-rays. So it's a complicated image analysis, but it gives you insight into the structure. So this allowed her and her colleagues to identify the molecule had a helical repeating sort of structure. It was linear, helical, and repeating. From her imagery and chemical work and Chargaff's rules and various other things, Watson and Crick, these two gentlemen here quite a bit later, or sorry, these two right here quite a bit later, excuse me, were able to identify that this was the actual structure of the DNA molecule, this double helix right here, with the nitrogenous bases facing in towards the center in pairs, and a continuous backbone of sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. That's what this blue strip right here represents. And following Chargaff's rules, they paired thiamine with adenine, and thiamine is a pyrimidine, a single ring molecule, so short. And adenine is a purine, a double ring molecule, so long. And so also we have T and A always bound to each other, a small molecule bound to a large molecule. G and C have the same sort of relationship. Cytosine is a pyrimidine, and guanine is a purine. So since uracil takes the place of thiamine in RNA, what would you expect uracil would be? Is it a purine or pyrimidine? Which one seems logical? So if you pause that and think about it and come up with an answer, I'll let you do that. There's a pause. Y'all can pause the recording. And let's go back and look. So uracil, like thiamine, is a pyrimidine. It takes on the same role, and it base pairs, of course, with adenine right here, small molecule to large molecule, or small molecule to large molecule. So that's all consistent right there. Some other details in the structure of the DNA molecule, as understood, and now, much confirmed, is that the base pairing cytosine to guanine has 3 hydrogen bonds. That's these dotted lines right here, bonding the 2 molecules together. So they are 33%, or sorry, they are 50% more tightly attached to each other than thiamine and adenine are. They've got 2 hydrogen bonds right here. These guys have 3. So, adding another one to 2 is a 50% increase right here. Or you could say, from the point of view of cytosine, the thiamine-adenine bond is only 66% as strong as the guanine-cytosine bond. So there's a little mini lesson in how you identify, ratios depending on your perspective. All right, and some other details about how the DNA molecule is put together. They're grouped together by these bonds right here. As it turns out, the strands are anti-parallel. So notice the ribose sugars right here. The oxygen is facing down on this side and facing up on this side. So one strand runs in one direction, the other strand runs in the other direction. And as we now know very, very clearly, polymerization, the building of a DNA molecule, proceeds from the 5 to the 3 direction. So if this is all the DNA we had right here, and we wanted to use the same mechanisms that a cell uses to elongate DNA, the next the next DNA monomer would be attached over on this end of this strand. We'd attach to the 3' carbon here. I'll define that in a minute. And we'd attach the next monomer, and so on and so on and so forth. So the 3' indicates the direction we can add pieces to. All right. Same thing over on this strand right here. Biologically, we can't add DNA to the 5' end right here. It only adds to the 3' end, so we build in that direction. So that means what we refer to as this structure is anti-parallel. The 2 strands are parallel to each other, but anti-parallel. They both have a tendency to bend in the same direction, but since they're anti-parallel, that complements, and that's what allows the molecule to wind up in the double helix. Finally, 2 other details. When this is twisted around itself, you have what's called a minor helix right here, where the 2 strands are a little closer together, and a major helix right here, where the 2 strands are a little further apart. And that that sequence repeats all the way. The significance of the minor and major grooves is that different classes of molecules attach to DNA and influence the behavior of DNA, including proteins and rarely other DNA, but quite frequently other RNA molecules. And it's more typical that proteins will attach to one of the grooves and in a non-specific way, so they don't base pair to monomers of DNA inside here. And in the other case, RNA molecules that do base pair, that a T binds to an A G binds to a C, that sort of base pairing, or U, binds to an A at uracil for RNA, can base pair to these guys, and we have different classes of molecules utilizing the narrow groove or the wide groove for that binding process. And so we have base pair recognizing binding of molecules to DNA. That means they can identify a particular sequence and attach to it. So that usually has very, very specific, that's a much more specific sort of recognition. And there's proteins that just generically bind to DNA and don't base pair to them. And that can happen over any portion of that helix. All right. Some very good experimental work was done by a number of different researchers and identified the mechanism for reproduction of DNA. Some details. The researchers that we most honor for their work on that were Misselson and Stahl right here, and they were working with a radioactive version of nitrogen, which is included in all nitrogenous bases, A, G, C, T, and U. And with a clever use of this radioactively labeled DNA, they were able to establish that the method of reproduction, and here's what they did. They had cells that were grown in a radioactive nitrogen source right here, so all of their DNA would be radioactive. And then those cells were transferred to a non-radioactive nitrogen source environment. And they continued duplicating their cells and duplicating their DNA. And what they found through some very clever fractionalization experiments, the radioactive heavy nitrogen is heavier, literally, than the non-radioactive nitrogen. It's a heavier isotope. It's a very careful screening process to separate the two. What they found was an answer to the question, is DNA replication a dispersive, a semi-conservative, or a conservative process? And I'll explain those. So in the conservative process, the double helix is used as a pattern to make a copy. So here would be the original, unaltered, and here would be the copy in a new cell. So there's old DNA and new DNA. The semi-conservative model takes it that each strand is used as a template to make its matching strand. So when you're done, you've got one cell, now you've got two cells, each with DNA in them. Each of those cells has half old DNA and half new DNA. And then finally, the dispersive model suggests that the DNA is chopped up into pieces, and those pieces are used as instructions for making the complementary portions in between the pieces. I think in this drawing right here, to reflect this ideally, these stripes should be alternating. So if there's a blue here, it should be gray here. If there's a gray here, it should be blue here. But that would be the dispersive method, and so that each strand of DNA is a mixture of old and new pieces, somehow copied, identified, and attached back together in the correct order. As it turns out, the semi-conservative model is correct, and this is how DNA replication actually occurs in all living cells, bacteria and eukaryotic. All right, next we want to look at the mechanics of actual DNA replication. So there's a series of 3 images right here. The first one is showing a strand of DNA being opened up, and this is called a replication bubble, just because it looks like a bubble, under very, very high resolution electron microscope where you can sort of see these things at work. So there's a replication bubble, and we've got the parent strand, both of the parent strands right here, and new DNA is being produced right here, the complementary strands. And these bubbles expand to the left and right. We have a replication fork moving to the left right here and to the right. Same thing here and here. And for a really long strand of DNA in eukaryotic cells, there are multiple replication bubbles occurring at the same time on a given DNA strand, and those bubbles get larger and larger until they connect to each other, and the result is we've got 2 complete replicate DNA molecules where before we had 1. And it says prokaryotes up here, but the multiple points of replication is true only for eukaryotic organisms. Prokaryotes just have a single replication point of origin, to our knowledge, for all prokaryotic cells, and that expands to the left and right until the 2 replication forks make it all the way around the circular chromosome and back, and join to each other and complete the process. But for the much larger linear DNA molecules that eukaryotic cells have, we have multiple points of origin of DNA replication. So those grow until they all join together, and we've duplicated the whole molecule. Looking a little bit more in detail right here, the directionality becomes really, really quite important. And there isn't, dang it, always an error in these pictures. Yes. No, they're getting on top of my recording. I can't have dogs barking in the back of the house. It gets, if you're going to go out.
I need pizza.
Okay. All right. I'm not.
Thank you, Bob.
So we're looking at this more detailed illustration of a replication bubble right here. Earlier there was some labeling over here that was incorrect, so discount that. And notice the 5 and 3' directions of the molecule right here. So this is the 5' end of the DNA molecule. So it would have been built in this direction, and this molecule would have been built in this direction. So because DNA is anti-parallel, the replicated material is anti-parallel. So 5 to 3, it's built in this direction right here. And over here, it's built in this direction right here. So all of the proteins involved in duplicating the DNA kind of cluster together. So we get this weird thing where we have a leading strand where once we start the replication process, we can continue it continuously right here and run all the way until we get to the end of the molecule or hit a previous site of duplication, like another replication bubble right here. But at the same time, we have to build short discontinuous bits of complementary DNA running backwards from our replication fork, which is proceeding in this direction. And that's what's called a lagging strand right here. And these fragments are called Okazaki fragments over the researcher who first identified them and characterized them. And so leading and lagging strands right here. But all of the duplication process utilizes the same enzymatic proteins to catalyze the reaction. And that's what we'll look at in the next couple of slides. So here's a slightly more detailed look at that replication fork right here. So here's the 3 to 5 strand, and here's the 5 to 3 strand right here. And so again, if we start duplicating somewhere, we can continue duplicating continuously in one direction right here as we unwind DNA and duplicate it. But in the other direction, we have to do it in these short little reverse bits. Do it here, do it again, and join these bits together. And those are called Okazaki fragments. All right. So here we're looking at the process a little more closely. And again, we see our 3 and 5' structures right here. And so here we have the leading strand running continuously, duplicating continuously, and here we have the lagging strands being duplicated in fragments over and over and over again. So you need to know the enzymes that are responsible for this process in their correct order. You need to know their names and their functions and the order in which they function. So taking it from the top, topoisomerase is required to unwind the inevitable twisting that will occur if you separate this helical molecule right here and then keep it separated into 2 new strands that then in turn twist up on their own. Imagine taking a rope or a big piece of string that's made out of 3 smaller strings that's wound together. And try pulling those 3 strands apart from one end without twisting and unraveling them. And you can do it for a little bit, but you'll get a big knot ahead of you, ahead of your pulling apart process. And at some point, it'll become unmanageable. Same thing for the DNA molecules. We pull it apart, that knot will occur. Topoisomerase cuts, unwinds the stress, and then rebinds the DNA molecule periodically to relieve that stress. So that's the task of topoisomerase. So it runs ahead of the replication fork, relieving strain periodically. Next is a molecule called helicase, which is kind of like a battering ram that pushes its way through and separates the DNA strands by breaking the hydrogen bonds between this would be a purine, and this would be a pyrimidine right here. But breaking the bonds between those nucleic acids. So that's the job of helicase right here. And then once the 2 molecules of DNA are separated from each other, to keep them from re-sticking to each other, because that's what they'll naturally do, those hydrogen bonds will reform, we have these proteins attaching to the DNA strand called single-strand binding proteins, which mechanically and through some charge processes keep the DNA from reattaching to itself. And those guys are taken off as we get into these duplication areas right here, and those proteins are reused upstream continuously. So they're recycled, recycled, recycled. Next, we have a enzyme called DNA primase, which produces an RNA primer. So what it does is it recognizes a particular piece of DNA, a particular sequence, and when it sees that sequence, it drops down an RNA version of that, an RNA uh, uh, opposite of that piece of DNA in the form of RNA, and that's what we mean by a primer. So it's made out of RNA. Once we have that primer in location, then DNA polymerase 3 is capable of attaching to that primer and reading the complementary sequence of DNA over here, or the template. Template just means the thing we're going to copy. And making DNA replicated material right here. All right, and so that's the job of DNA polymerase 3. And then DNA polymerase 1 is another version of this enzyme. So we've got DNA polymerase, this should be 3 right here. This should be DNA polymerase 3, and here's DNA poly 1. DNA poly 1 replaces the RNA primers with DNA, replaces those guys. And then whenever a duplicated bit of DNA hits a previous primer, poly 3 DNA poly 1 will replace the primer with the RNA primer with DNA, and then ligase will bind the two pieces together, and that process repeats continuously on the lagging strand. And it only occurs on the leading strand when this replication fork right here catches up with a previously initiated point of duplication, like this bubble expanding until it hits this bubble right here, and then we'll end up with the same sort of situation occurring. So anyway, those are the enzymes involved in the DNA replication process. So let's go over them again. A replication fork is formed when helicase separates DNA strands. Up ahead of DNA helicase is going to be topoisomerase, which undoes that overwinding by cutting it, cutting one strand of DNA, allowing it to spin around the other one, which stays connected. All these are single covalent bonds, so they're all rotating bonds in the backbones of the DNA molecule. And then we hold that replication fork open with those single-stranded binding proteins that get used again and again and again and again and again. They're all reused. DNA primase recognizes, RNA primase, recognizes a particular sequence of DNA, and whenever it sees that sequence, it drops a primer down. Polymerase 3 attaches to that primer and uses it as a starting point to build replicated DNA. So at the beginning of your replicated bit is going to be a little piece of RNA, and then the rest of it will be made out of DNA. DNA polymerase 1 replaces RNA with DNA in that primer, and then DNA ligase is the last actor in this process, and it joins together those components of the lagging strand, the Okazaki fragments, putting them together to make a continuous strand of material. So that ligase has to be used a lot on these lagging strand portions right here, joining each of these together. This dark spot right here represents a primer. The light represents, DNA replacement. And then we have to replace that RNA primer with DNA using DNA polymerase 1, and then join these arrows together using ligase to join them together. Ligate is the fancy Latin term for sewing. A ligation is a fancy term for a stitch in medicine. All right. Let me go back and let's remind you about this 5-2-3 structure right here. We've highlighted it quite a bit, and notice it keeps showing up, and remember the strands are always anti-parallel to each other. So I'm going to go all the way back and let's take a look at the basis for that structure. So here we've got a ribose sugar right here, and this is where the 3 and 5 come from. So by convention, we count carbons starting on the simpler part of the sugar. So remember what this means right here. The lines represent hydrocarbon, and anything that's not hydrocarbon, hydrogen and carbon, we go ahead and label in these models. So there's some oxygen and nitrogens and phosphorus is all labeled because they're not carbon or hydrogen. So if there's nothing noted, it's carbon and hydrogen. So here's a carbon. That's carbon number 1. Here's a carbon. That's carbon number 2. Here's a carbon. That's carbon number 3. That's where the 3 identity comes from in this direction. Here's carbon number 4. Out here is carbon number 5. It's a pentose sugar. 1, 2, 3, 4, 5. Here's the 5th sugar. Organic chemists insist this is the way we count. So we do 1, 2, 3, 3 prime, 4, 5, 5 prime. That's how the directionality goes. Try it yourself over here. Remember this over here, the complicated carbon furthest away on the chain is considered the last one. The simplest carbon closest in is the first one. So here's carbon number 1. You do the counting this time. All right, so we've talked about replication. Let's talk about a couple of little details that come in here. So mistakes occur in replication because it's a really rapid, fast process. It's going extraordinarily quickly in real time, and the onus, the imperative of the cell is to get this duplication done very quickly because while the DNA is all pulled apart and being duplicated, none of the genes can be read, no proteins can be synthesized. The cell is basically kind of in a zombie pause. It can't adjust anything it's doing to respond to changes in its environment. And if that persists, the cell's going to become unstable and die. So it has to get the DNA replicating process done quite quickly. And so kind of the cost of that rapid replication is the appearance of errors periodically. There are also errors introduced into DNA through external factors, radiation, carcinogenic chemical compounds, which basically damage or break DNA, and then the DNA replicating enzymes that we've already looked at are recruited to repair and reconnect the damaged DNA. So this happens all the time in living cells. Bits of chemical damage to DNA are repaired all the time. So we've got a proofreading mechanism, all right? And so as we make a duplication of an existing strand right here, periodically we'll make an error. So here we've got a guanine that's been introduced instead of an adenine right here. So for whatever reasons, DNA polymerase introduced this error right here. G does not pair with A, we know that, okay? What's going to happen is when we've got that mismatch, G and T in this particular case, and a G and an A in an earlier example, won't match with each other. What's going to happen is we, in the daughter strand, will go ahead and identify the base that's not matching. In this case, there's a thiamine right here that's not matching. And so we cut it out and we'll replace it with a cytosine right here. And now we've corrected the error. How does the cell know which thing to correct? I mean, we could have taken the G right here and replaced it with an adenine to match the T. But remember, blue in this particular picture right here represents the old original DNA, and green represents the new DNA. So the proofreading and the mismatching repair mechanisms normally use the older strand of DNA as the model and use it to make the new strand of DNA. And then if there's a mismatch between them, inevitably it's the new strand that gets corrected to match the old strand if the mismatch is identified and fixed. So that's the beauty of the semi-conservative model. Let's go back and take a look all the way right here. Of this, right here, every new strand of DNA has an old, template that is used as the, as the basis for making the new complementary strand right here of a double helix. And if there's a difference between these two, chemically this is recognized as being the old DNA molecule, so the corrections are always made to the new molecule. That's the beauty of the semi-conservative process. Every cell has a template to, to double check its new DNA against, as it goes forward. All right. So talking about repairing errors, so there's some examples right now. And so we've replaced that, particular erroneous bit of DNA. All right. There's also a phenomenon where two thiamines, when they're next to each other, periodically base pair to each other. They hydrogen bond to each other. They cross bond to each other, which makes them incapable of binding to their complementary adenines over on the other strand. And so there is a mechanism, and there's a number of these where, something that's showing some irregularity like this, we've got enzymes that can take out a whole big chunk of DNA and replace that material. So that's And then this particular case, this is a well-known error where these thiamines bind to each other. It's usually in a high energy state when we've got UV radiation, this thiamine dimer right here. So there's a mechanism for replacing that as well. All right. A very interesting phenomenon when we duplicate our DNA because of needing to have a primer as a starting spot for DNA replication. There's no rule that says the pattern that RNA primase recognizes is the exact end of the DNA molecule. So the first primer that shows up might be a little ways in from the end of the molecule. And secondly, if we have a primer, DNA polymerase 1 can only start with existing DNA and then replace a primer with DNA running in the 5 to 3 direction. So if there's a primer way off at the end of a DNA molecule right here, there's no way to replace that primer with DNA, that RNA primer with DNA. So the long and short is, as we duplicate chromosomes in an alternating little stepwise sequence right here, we lose and lose and lose chunks of the DNA molecule. Replication continuously shortens the ends of eukaryotic linear chromosomes. This is one of the mechanisms that's understood to contribute towards cell death, and if we have enough cells dying simultaneously, organismal death in a multicellular organism. Chromosome shortening. So cells that we identify as stem cells, those cells that we call stem cells, have a gene or a series of genes, but a key gene that is functioning, which produces an enzyme called telomerase. And telo means the end or extreme, boundary of something. So telomerase is an enzyme that can recreate the missing shortened section of the DNA molecule and rebuild that. So it re-extends the telomeres of eukaryotic linear chromosomes. So cells that are stem cells, which means they can divide for an unlimited number of times, can do that because their DNA molecules don't shorten, and that is because the telomerase gene is active, synthesizing the enzyme telomerase, which makes telomeres. It builds the ends of these chromosomes out, and that's an immortal cell. A cell that is expressing its telomerase gene is an immortal cell, and that's what we mean when we say a stem cell. A stem cell is a cell that can divide to produce daughter cells indefinitely. Elizabeth Blackburn got her prize in 2009. The work was much, much earlier. Typically, there's about a 10-year lag right here. I don't know the date offhand, but you can look it up. So she did her work identifying how these telomeres work, which was a very, very important piece of information. That's why stem cells have been such an important and controversial subject. Classically, stem cells are present in in a developing embryonic animal. For instance, in us, we have stem cells. All of our cells are stem cells technically early on during our embryonic development. And then as we mature, more and more of our cells stop being stem cells and become dedicated tissue-specific cells that have a limited number of replacements ahead of them. And that's one of the things that causes us to age and, grow frail as we get older and older. So identifying how telomerase works was an important step in coming up with something now. Now we can take cells from any part of your body or any organism's body, so any animal this can be done to. We can take any normal cell that is subject to this situation right here, reactivate its telomerase gene, get it to express telomerase, and basically force a skin cell from you to become a stem cell. So a skin cell or a muscle cell or whatever, but a skin cell is a typical target. And turn that back into a universal stem cell for you that can make tissues and cells for you indefinitely. So now we don't have to go back to an embryonic source to get these stem cells. We can make them ourselves from the adult cells of an adult individual, and therefore use that as the basis of growing new tissue for that individual. That is a burgeoning area of regenerative medicine. All right, a little bit on errors and duplications that are not corrected. All right, so there's a couple of ways in which errors occur, and they have some consequences. So we'll look ahead at it in the next chapters, but the DNA code, A, G, C, T, etc., etc., is a alphabet, of 4 letters that are grouped together into 3-letter words, and these 3-letter words code for a particular amino acid. So here's serine, here's valine, here's proline, here's tyrosine. So A, G, C in that order is the code for serine, etc., etc., etc. So that's the DNA code right there. So significantly, if we swap out a letter for another letter, we change an amino acid. All right. So in this particular case right here, we've replaced an A with a T right here. Okay. Well, as it turns out in the dictionary, there are at least two words that code for valine. GTA, and this is 3, 2, 5. This is the 3 end of the molecule. This is the 5 end of the molecule. So GTA written in that direction codes for valine. GTT also codes for valine. All right. Now, here's another situation right here. In this sequence right here, the C, the leading C on proline, CCC, is replaced by an A, all right? And ACC does not code for proline, it codes for threonine. So we've replaced this amino acid with a completely different amino acid right here. And if you'll go back to the amino acid dictionary that we learned, or the list of amino acids that we learned some time ago, we can, look at the chemical nature of these 2 amino acids right here. And for instance, if this one, and I never remember this, you can look it up, but if proline happened to be, all right, and the third point mutation error. Point mutations means where we change one letter into a different letter. So here we changed a C into a T, here we changed a C into an A, and here we're changing a C into a G. And in this case, it's called a nonsense mutation because it introduces a stop codon in the language of codons, which is a set of 3 letters in DNA. Each codon has a meaning. There is one codon that codes for an amino acid and starts the coding process. So the first amino acid is always methionine in the initial translation product because of the start codon AUG. Then all the other codons mean some amino acid except for 3 different codons, which mean stop, and they code for no amino acid at all. And one of those stop codons is TAG. It means no amino acid, stop the translation here. So what means is when we change a codon that codes for an amino acid to a stop signal, we've made, at the very least, a protein with 1 less amino acid on it than it should have had. And if we introduce that stop really, really early, where there should have been 1,000 amino acids in a row, and we introduce a new stop codon by making this error at, say, position 20, the 20th amino acid, suddenly we've got a protein with only 20 amino acids instead of 1,000, and you can bet it won't do its original job, not even close, whatever that was, and that could be a pretty major error. So these are all point mutations. We can replace a letter and it doesn't change the meaning. That's a silent mutation. A missense, where we change the letter and we change from one amino acid to another amino acid with the possibilities that entails. Here's an example right here. If we go from a nonpolar amino acid to a polar amino acid, or an acidic or a basic, sorry, this is a basic and these are acidic amino acids right here, we could really alter the structure of the protein in question. Like right here, let's see, what did we do? We changed proline to threonine. So let's see what that's going to end up doing. Proline to threonine. Let me see if I can find there we go. Proline to threonine. So there's proline. It is a polar uncharged R group right here, and we changed it to threonine. which is another polar uncharged group. So in this particular case, the replacement wasn't too substantial. There's a difference in shape right here, so it may have effects. It might slightly alter the shape of an active site on an enzyme, or slightly, change the body of the enzyme. It wouldn't be as dramatic as if we switched from, say, polar to nonpolar, or polar to acidic, or polar to basic right here. That would have a much bigger impact. Or switched out that relatively small R group right here for a really huge R group like tryptophan right here. Those would have much more substantial effects on the structure of the protein. All right, so here's an example where a point mutation has a much more dramatic effect. So here's the DNA sequence, some of it that is responsible for the hemoglobin molecule that you find in red blood cells. And there's a sequence right here that includes glutamic acid, glutamic acid or glutamate. All right, and that is I'll show you right here. Glutamate is a polar molecule right here. So it's got a polar group right here. It dissolves in water. It's hydrophilic, and it's going to have a particular effect on the shape of and behavior of the protein. We replace that with valine, which is a nonpolar, non-charged amino acid, and the result of that single change, a one-letter change, which changes one amino acid to another out of a fairly large molecule with a whole bunch of amino acids, is that we go from regular red blood cells to red blood cells that have this sickling defect. The hemoglobin actually polymerizes into a super long filament, stretching the red blood cell that contains hemoglobin under low oxygen pressure conditions, which is like when you're exercising really, really hard. This happens to the red blood cells as they leave the capillaries and enter the venous system. And then they cause clogs somewhere downstream in the next capillary bed they hit. Can be quite damaging or even deadly. A one-letter change. So you can see that that's a pretty substantial effect. All right, and the last error that we want to talk about is a frame shift mutation. And this occurs when you insert or delete, most simply, a letter. You add an extra letter or you take out a letter from a sequence and you change the meaning of everything. All the codons downstream of that insertion or deletion have all been changed. And that can be a really, really terrific amount of damage to the protein structure. If it happens very early on in the sequence that codes for a string of amino acids early in the polypeptide, the whole protein can be utterly different. The same things happen when we relocate pieces of DNA from one location to another, or flip them back and forth, or any number of other large-scale errors that occur during DNA replication, DNA duplication. All of these things can occur. So a frame shift mutation. Here's an example. It's kind of silly, but it illustrates the point. So here we've got a series of codons that have meaning. Cat ate the dog. But if we add one extra A into the sequence, we frame shift everything over, and now none of the other codons have any meaning in this context. In DNA, every three-letter codon means something. It either codes for an amino acid, start, or a stop codon. They all have meanings, but they may not have useful meanings. These could be words in some other language, but they don't have anything to do with, our English sentence up here. So that's an example of a frame shift mutation. These are really, really usually quite deleterious, which is a fancy way of saying bad in a genetic sense. All right. I encourage you, this is a long film, so I won't play it on our recording right here, but I do encourage you to watch this. This is great. It's really, really good. Looking at, human history and taking a look at both the Neanderthal and the Denisovan genomes. Denisovans are another group of archaic relatives of the of Homo sapiens, so Homo neanderthalis, Homo denisovus are the two other related species in the Homo genus. There's a couple of others that are extinct as well, but we recognize now through some interesting research that he synopsizes that the Neanderthal DNA, there is a fraction of that in certain populations of humans. So there's also a fraction of Denisovan DNA in certain human populations as well. And it's really quite fascinating. So I strongly encourage you to watch this TED Talks. It is a TED Talk. All right, some last ideas right here. Y'all know this. We've gone over this quite a lot. Here's a eukaryotic cell, large and complex with lots of internal compartments. Here's a prokaryotic cell, eubacteria or archaebacteria, a smaller cellular organism without the internal compartments, without the complicated structure and all of that sort of stuff. When we're talking about DNA, the eukaryote has many large linear chromosomes. The particular number varies from species to species, packed inside a compartment, the nucleus. But the key feature is that the DNA is very large. There's a whole lot of it, and it is linear. It's strands that have a beginning and an end. In a prokaryotic cell, the DNA is typically one single chromosome that is a loop. It's folded up tight. In this particular case, they've drawn it badly. It shows, you know, ends at both ends here, but that's pretty atypical. It's usually a loop. Imagine taking a string, tying it together so it makes a loop, and then crunching that loop up, you know, so you can pack it into a small space. And that's what prokaryotic DNA typically looks like. And it's in a region within the cytoplasm of the cell called the nucleoid, where there's a whole lot of proteins present that are going to manipulate that DNA. All right, in eukaryotic cells, it's important to recognize the structures that allow for packing and unpacking of DNA, because this turns out to be important in DNA regulation, expression regulation. So here's the double helix itself, all right? And inevitably, that double helix is wound twice. There's actually two loops. That's what we're looking at here, around a very complex protein structure called a histone, which consists of, if you count them right here, 8 individual unique proteins, globular proteins that make a great big ball, if you will. And the DNA is double wrapped around those histones over and over. And those histones are able to attach and detach from each other to clump the DNA together or relax the DNA. And so that gives you the chromatin fiber, which is the normal state of DNA in all cases everywhere in our cells. And then if those histones start aggregating together and then clumping together with other proteins that pack themselves in here as well, we get condensed DNA that superfolds on itself into a tighter and tighter structure, ultimately until it's, you know, compacted all the way down into this super compact chromosome, which is typically the way we see it after DNA duplication during the prophase and certainly metaphase stages of the cell cycle of DNA, cell division, when the chromosomes have to be compacted so that they can be moved from one location to another. And a key actor in that compacting of the DNA is the histone proteins that DNA is wrapped around. So it doesn't actually look like this straight out inside our cells in vivo, which means, you know, inside our cells when they're alive. All right. A little bit about DNA sequencing. This is pretty good. There's a short animation, again, that y'all need to watch. We don't need to review it in this recorded video right here. But the Sanger method of DNA sequencing. So what you do is, you remember PCR. We did PCR in lab earlier on in the semester before the break. The technique for PCR, you modify it a little bit by throwing into your mixture, along with good old regular adenine, you know, triple phosphorylated A's, G's, T's, and C's. Along with those, we throw in special fluorescently tagged versions of those A's, G's, T's, and C's that can't be added onto in the 3' position. They're called chain termination nucleotides, and they're fluorescently tagged or radioactively tagged. It used to be radioactive, now it's fluorescent. So when you shine a UV light on them, they fluoresce in a particular color, and it's, as you see here, black, green, red, and blue in this particular illustration. I don't know how you're fluorescent black, but there it is. And so what happens is, when you're, you know, making a copy of your DNA, it'll be so many bases long and then suddenly stop, you know, with a radioactively tagged C or a fluorescing C right here, and stops. And so, you know, position 20 is a C, and position 19 down here is a G, and position 18 here is an A. We get a whole bunch of these, you know, this, and so on and so forth. And that's the technique that's used to generate a a DNA sequence. That's how we actually sequence DNA, or at least one of the mechanisms. There's some faster, more exotic versions that are used now instead of this, but this was the first one that gave us the ability to read a whole lot of DNA pretty quickly, and it's called the Sanger sequencing dideoxynucleotide, and so it's the dideoxy version that you can't add another nucleotide to, and so it causes the chain to stop with a fluorescent tag at the end, because the dideoxynucleotide is also fluorescently tagged. Here's an excellent video showing it. In fact, I will play it because it's not very long. Let's go ahead and do it.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into two strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
So as I said, that's nice and short.
The first method of sequencing the genetic code was devised by Fred Sanger.
Sorry about that. I didn't realize it had gone on to loop. So that was a short but very, very illustrative, little video of the key feature of the Sanger method of DNA replication. All right. You're also familiar with DNA electrophoresis, so we can identify fragments of DNA. We don't know the letters, but we can know the size of the piece of DNA, which is normally called the number of base pairs. An A plus a T on the other side is a base pair. A G plus its corresponding C on the other side is a base pair. And we can, separate chunks of DNA just like we can proteins based on molecular size, not the sequence of letters, but the molecular size. And then if we happen to have a ladder, which is, a pile of known pieces of DNA, grouped together in particular, sets, and know the sizes of them, we can estimate the number of base pairs in a chunk of DNA. And that turns out to be a very useful technique for a whole bunch of things that we get done with DNA information. All right, so that's the end of the slideshow. I do want to show you one last animation that we'll conclude with, and I think you'll find this one really rewarding. All right, so I'm playing this. This is available on YouTube. You can, I think, figure out its identity right here. Let's go ahead and play it. So there's the histone proteins that DNA is wrapped around. We'll study this in the next chapter, transcription and translation. But we're looking for the DNA replication portion, which I thought this was the right video. I'm going to have to go back. Here we go. I found it. Well, this is a whole lot of good stuff. Drew Berry's animations of cell structure, nucleus exterior, cell cycle. Let's skip ahead right here. Alright, we're about to start on DNA replication. So we'll show an animation of DNA replication theoretically at real time. So let me stop. What's going on right here? So we've got the double helix, the original strand of DNA coming in right here, and it is being duplicated. It's split apart and duplicated into, one duplicated and the other duplicated. So remember, it's semi-conservative. So this thing, half of this is going down this pathway and half of it is going down this pathway. And then we're synthesizing the complementary bases on, on the other half of the strand. And remember, there's a leading strand that's continuously replicated in this direction. That's going to be the leading strand. And then we've got this lagging strand, which has to be done in these loops where duplication actually runs backwards. And then there's a mechanism for kind of including those together. And that's what we're looking at here. And so this complex right here of all of these proteins includes topoisomerase and helicase and primase and DNA polymerase 1 and DNA polymerase 2 and ligase. Those are the enzymes that you need to know in order. Oh, I forgot. Let's include the single-stranded binding proteins. So in order, topoisomerase, helicase, single-stranded binding proteins, primase, DNA polymerase 3, DNA polymerase 1, ligase. That's it. So let's watch. So you can see the lagging strand being replicated in pieces. And here we're looking at it a little more closely. And it's frightfully fast. So this is considered to be at real-time speed based on, there's some ways of establishing how quickly this happens. And the way that's figured out. All right, so this is transcription and translation. We'll save that for another day. But the way they figured out the speed at which DNA, that complex is replicating is by knowing the size of a genome, say, the 3 billion base pairs that makes up your DNA, and under electron microscopy, seeing on average how many replication bubbles there are, therefore locations, and each bubble represents two, duplication events, one going to the left, one going to the right. How many bubbles there are on a given chromosome, and knowing the size of that chromosome, and then if you do the math, you can figure out, and how long it takes for the cell to duplicate its DNA. In other words, how long does it spend in the S phase of the cell cycle, and that can be established experimentally. While doing all of that, you can calculate how many base pairs a second need to be duplicated to get it all done in the time observed, and that's how that estimate was made. So that's it. That's the chapter on, and I'm sorry this was a bit longer of a video than normal, but that is the fascinating, in my opinion, chapter on DNA structure and replication, with some considerations of the history of DNA replication and some considerations on the effects of errors in DNA replication, both important.